So if you see the male reproductive system, it is mainly divisible into two parts. What are those two parts? First is the male reproductive organs and then male external genitalia. Okay, reproductive organs and external genitalia. So here you can see the list of the male reproductive organs and then here the external genitalia. Okay, now in histology we don't have to study everything. There are some important selective topics or points which we have to discuss. Okay, today I will try to complete as much as possible. Whatever will be left we will discuss during the histo demonstration class. Now if you see the list, first testis is there, okay, then epididymis, ductus deferens, seminal vesicles, ejaculatory duct, prostate gland and bulbourethral glands. Okay, prostate glands, bulbourethral glands, and in case of external genitalia, first penis is there, urethra and scrotum. Okay, now have you studied about the testis, gross anatomy of testis? Okay, very good. So I hope you have seen the testis, the structure of the testis, everything. So today we will discuss few important aspect which is related to the gross anatomy, and then I will start the histology. Now in this particular diagram, can you see the parts of the testis? Anyone, do you know the parts of testis? You have studied, so you should know the parts of testis. Okay. I don't know whether you have done this in dissection or not. There is a point called site identification of the testis. Site identification of the testis. Have you studied that? When it is in the body, it is very easy to identify which is right testis, which is left testis. Suppose it is taken out of the body, okay, and then if you have to identify the site, those questions are asked especially during the time of practical exam. Do you know how to identify which side of the testis, right side, left side? Okay. So I hope that point is left. Better you study during the time of practical classes, especially during the time of dissection, how to identify which is right testis, which is left testis. It can be done. Okay, the way you have studied about the identification of bones, in the similar way you can identify the site of the testis. Okay. Okay, so forget about that. Now we are not going to discuss. Okay, I thought you have studied, that's why I am asking. Now in the we know that two testes are there. Okay, so everything related to the testis are two. Please keep that in mind. Okay, so two testis, everything. I am talking about external feature. Everything is two. For example, if I ask you what are the parts of testis externally, what will be your answer? It is having two poles, upper pole, lower pole. Okay, then two borders are there, anterior border, posterior border, and two surfaces are there, medial surface and lateral surface. Okay, so everything related to the testis are two. Okay, so two testis, everything is two. Okay, so use all those methods in order to understand it properly. Means it will be easy for you to understand. Okay. Now in this particular diagram, you can see various parts of the testis. Can anyone identify which is the upper pole? Yes, where you can see the epididymis is attached. That is the upper pole. Okay. So originally, if this is the upper pole, then this will be the lower pole of the testis. Okay. Next, here you can see this is the anterior border of the testis, and this is the posterior border of the testis. This is the medial surface, and on the other side there will be the lateral surface. Okay. So like this, these are the various parts of the testis. Now, after that, in histology, okay, if you see, the testis is covered by three coverings. 3 layers of coverings are provided to the testis, if you have studied. Okay. So today we will see in brief about those coverings, because those coverings are also important. Unless and until you understand the covering, you cannot understand the internal structure. Okay. So here you can see the coverings of the testis. So as I said, it is having 3 layers of covering from outside to inside. So from outside to inwards, what are the layers? First is the tunica vaginalis, tunica albuginea, and then tunica vasculosa. These are the three layers of coverings as you can see in the diagram. Okay, so first if you focus on the diagram, here you can see the layers. The first is the outermost is the tunica vaginalis. I am talking about only about the testis. Now if you consider the testis which is suspended in the scrotal sac, at that time the number of coverings will increase. For example, you will be getting the skin, you will be getting the external spermatic fascia, you will be getting the cremasteric fascia, internal spermatic fascia. All those layers will come when you will include the scrotum. Okay, if you exclude the scrotum and if you see only the layers of testis, then three layers are there. Okay, now which is the outermost? First is the tunica vaginalis. Okay, now please keep that in mind. Tunica vaginalis, the layer tunica vaginalis, it is again divided into two different layers, parietal layer and visceral layer. Now your question will be, why here parietal layer and visceral layer is coming? Okay, if you can see the diagram, tunica vaginalis is being divided into two layers. Outer one is the parietal layer, inner one is the visceral layer, separated by a small cavity. Have you studied the descent of testis? So when you have studied the descent of testis, you should know why two layers are there in the tunica vaginalis. Because the processus vaginalis, which helps in descent of the testis, okay, the testis will invaginate into the processus vaginalis. Okay, as a result, it is having two layers. Outer one is the parietal layer, inner one is the visceral layer. Okay, so here also we are having two different layers in case of tunica vaginalis. Inside that, you can see the layer which is present is the tunica albuginea, which is made up of fibrous tissue, dense fibrous tissue. It is also performing a very important role. Okay, because have you heard about the mediastinum testis? If not, okay, if you have not heard about the mediastinum testis, please keep that in mind. The tunica albuginea, fibrous layer, okay, which is covering the testis, okay, it send a vertical septum, incomplete vertical septum from the posterior aspect of the testis. Okay, so that particular vertical septum is forming the mediastinum testis, which is very important. And from that mediastinum testis, the various septa will go and divide the testis into various lobules, as you can see in the diagram. Okay, so I am talking about this mediastinum testis. From mediastinum testis, you can see the various lobules of the testis are formed, and inside that lobule, seminiferous tubules are present. Okay, so tunica albuginea is there. Okay, as you can see, tunica albuginea. So tunica albuginea is a fibrous layer, and inside that we are having the tunica vasculosa, where blood vessels are present. Okay, so like this, three layers are present in the testis, and the outermost layer, as I said, it is having two different layers. It is outer one is the parietal layer, inner one is the visceral layer. Okay. Now tunica vaginalis is the lower persistent part of the processus vaginalis, okay, which helps in the descent of the testis. Okay, now as I said, the testis is invaginated, okay, from behind, and therefore has a parietal layer and a visceral layer, and in between them there will be a cavity. Okay, as you can see in the tunica vaginalis, it is representing the lower persistent portion of the processus vaginalis. Okay, now if you see the tunica vaginalis, it is covering the whole testis except the posterior border. Now you will see mostly the posterior border is not covered by the layers. Can you tell me why most of the layers are deficient posteriorly? Okay, can you tell me why? Let me show you the diagram. Now see, this is in this particular part. Okay, most of the layers are not present in this particular part. Why? Yes, because blood vessels has to enter. Okay, how it will enter? Through that posterior border only. That's why most of the layers are absent in that particular area. Next, tunica albuginea. So, as I say, tunica albuginea is a white fibrous coat that is covering all around the testis. Now, it is covered by visceral layer. So, tunica albuginea internally or inner part of the tunica albuginea is covered by visceral layer of the tunica vaginalis. Okay. Now, except posteriorly, why posteriorly it is not present? Because the blood vessel and nerves has to enter the gland. Okay, and the innermost layer is the tunica vasculosa, vascular coat of the testis lining the lobules. Okay, so just now I have shown you. See, these are the lobules. So inside the lobules, a vascular coat is there, where the blood vessels are present. That is the tunica vasculosa. So like this, we are having three layers. Okay, covering the testis. Now, please see the diagram carefully. Can you see all the layers? See, tunica vaginalis, two layers are there. In between them, a cavity is present. Okay. Next, you can see the tunica albuginea, which is a dense fibrous layer. And after that, you can see tunica vasculosa. Okay, the vascular layer, which is mainly made up of blood vessel. Now, this tunica vasculosa is mainly lining or mainly covering the lobules of the testis. Okay. So, see, these are the lobules of the testis. Okay, these are the lobules of the testis. Inside this lobule, what are present? What is present inside these lobules? Seminiferous tubules. Okay, so inside these lobules, what is present? Seminiferous tubule. And we have to concentrate in this particular seminiferous tubule when we are discussing about the histology of testis. Now, if you see the structure of the testis, so the tunica albuginea, which is there, okay, it is a thick fibrous membrane. First of all, it will cover the entire testis and project into the interior of the posterior border as an incomplete partition that is called as mediastinum testis. Okay, I have shown you just now in the diagram. So from the covering, that is the tunica albuginea, which is a thick fibrous membrane, it will project into the interior of the testis from the posterior border as a incomplete partition, and that is called as mediastinum testis. Okay. Now, from the anterior surface of the mediastinum testis, numerous septa will radiate and will divide the testis into various lobules. Approximately 200 to 300 lobules are present inside the testis. Now, if you take each particular lobule, what is the shape? It is conical in shape. Each particular lobule is conical in shape, and if you see the apex, it is directed towards the mediastinum testis. Okay. So, the fibrous membrane, that is the tunica albuginea, which covers the entire testis, project into the interior from the posterior border. Okay, in an incomplete partition, and that is called as mediastinum testis. From mediastinum testis, numerous septa will radiate and divide the substance of the testis into numerous lobule. If you take one particular lobule, what is the shape of that lobule? It is conical in shape, and the apex is directed towards the mediastinum testis. Okay, so those lobules will constitute the glandular part of the testis. Approximately 200 to 300 lobules are there. Inside each lobule, what we are getting? We are getting 2 to 3 highly coiled seminiferous tubules are present. Okay. When we will stretch these particular lobules, it will measure approximately 60 centimeter. Just imagine, inside the each lobule, highly coiled seminiferous tubules are there. 2 to 3 seminiferous tubules, highly coiled. Now, if you stretch the coiled part, the length will become up to 60 centimeter. That is the length. Okay, and diameter is very less, approximately 0.2 millimeter. Okay, so I am talking about this mediastinum testis. This is the mediastinum testis. So from mediastinum testis, you can see the septa is radiating. Okay, and it is dividing the testis into smaller lobules. So see, this is one lobule, this is one lobule, this is one lobule. What is the shape of the lobule? Conical in shape. The apex is directed towards the mediastinum testis. Okay, what is present inside the lobule? What is present? You can see these are the highly coiled seminiferous tubules. Okay, 2 to 3 seminiferous tubule inside each lobule. If you stretch it, the length becomes approximately 60 centimeter. What will be the diameter? 0.2 millimeter. Okay. So now we are inside the lobule, and that particular lobule is forming the secretory part of the testis, or you can say parenchyma of the testis. Okay. Next, so those seminiferous tubules which are there, okay, what will happen? What they will form? So the seminiferous tubule, first of all, they will join together at their apices, means at the apex of the lobule. This seminiferous tubule will join together to form 20 to 30 straight tubules. Okay. Next, they will anastomose with each other to form a network of tubules that is called as rete testis. Okay. After that, the rete testis will give rise to 10 to 20 efferent ductules which emerge near the upper pole of the testis, and then they will enter into the epididymis. Those 12 to 30 efferent ductules, okay, ultimately they will form a highly coiled tube. Those highly coiled tubule will form the body and the tail of the epididymis. Okay, and then the epididymis will continue as vas deferens. Okay. As you can see in this diagram, okay, can you see the mediastinum testis? So see, this is the mediastinum testis. After that, these are the lobules. You can see these are the lobules. Inside the lobules, I hope you can see the seminiferous tubules, highly coiled. Okay. Now, this is the apical part. This is the apical part. This is the apical part of the lobule. So, near the apical part of the lobule, what will happen? The straight tubules will be formed. Those seminiferous tubules which are there at the apical part of the lobule, they will join together to form straight tubules. Approximately 20 to 30 straight tubules are there. Okay? Now those straight tubules, what will happen? They will anastomose with each other. Okay? They will anastomose with each other to form a network that is called as rete testis. As you can see, this is the rete testis. Now from this rete testis, see, we are getting approximately 12 or 15 to 30 efferent ductules. Now those efferent ductules will emerge at the upper pole of the testis. Okay, as you can see, this is the upper pole of the testis. Okay, and then they will enter the epididymis. Now these efferent ductules, okay, they will constitute mainly the head part of the epididymis. After that, this straight efferent ductules, okay, they will form a highly coiled tube. Okay, highly coiled tube that will constitute the body and the tail of the epididymis. As you can see, this is the body and the tail of the epididymis. After that, it will continue as vas deferens. Okay, so this is the direction how the spermatozoa is traveling through the various part. Okay, it is the conducting part, as you can see. Now what we have to, this is the normal structure. Next, we'll go inside the seminiferous tubule. Okay, so I have shown you where the seminiferous tubules are present. Okay, seminiferous tubule is continuing as what? Okay, ultimately we are getting the epididymis and vas deferens. Now if you go inside the seminiferous tubule, what are the different types of cells you can expect? Now, if you see the structure of the testis, first of all, 3 coverings are there, which already I have shown you. As you can see, tunica albuginea, tunica vasculosa, and tunica vaginalis, then tunica albuginea, tunica vasculosa. Okay. After that, inside that we are having incomplete lobules. The lobules are not complete; it is incomplete, but it is conical shape. Next, the seminiferous tubules which are there. Just now I have shown you the seminiferous tubule. So inside the seminiferous tubule, mainly 2 types of cells are present. What are those cells? As you can see, the spermatogenic cells are there, where we are getting the spermatogonia, spermatocytes, spermatids, and spermatozoa. I hope you have studied the stages of the development. Spermatogenesis you have studied, right? Spermiogenesis also you have studied. So you know what are the different stages of the spermatogenic cells. Okay. Another important cells which are present inside the seminiferous tubules, these are the Sertoli cells. Okay, these are the Sertoli cells. They are having very important function. Sertoli cells, mainly we know that these are the supporting cells for the spermatogonium. Okay, so they are large pyramidal cells supporting role. Okay, they also removes the residual body which is present inside the lumen of the seminiferous tubule. Next, another important types of cells are there, that is called interstitial cells of Leydig or Leydig cells. Now those cells are polygonal cells having round nucleus lie in a cluster in between the seminiferous tubule. Now please keep that in mind. The interstitial cells of the Leydig, okay, they are not present inside the seminiferous tubules. Okay, so seminiferous tubules are there. Okay, in between the seminiferous tubule, the connective tissue part, in that part only these Leydig cells are present. Okay. Next, if you see the straight tubules and rete testis, okay, these are also the tubular part. So in case of straight tubules, it is lined by Sertoli cells. So inside the straight tubules, Sertoli cells are present. But if you go to the rete testis, they are lined by cuboidal to columnar cells. Now in the microscopic structure, so till now whatever we have discussed, those that was not microscopic part, because that can be seen normally if you take a nice section of the testis. Next, if you want to see what is present inside the seminiferous tubule, okay, at that time microscopic structure is required. Okay, so inside the seminiferous tubule, what you can expect, two types of cells are there. Okay, what are those two types of cells? As you can see, Sertoli cells are there and spermatogenic cells are present inside the seminiferous tubule. Okay, so if you see the seminiferous tubule, it is lined by complex stratified epithelium called seminiferous epithelium, or also called as germinal epithelium. Okay, inside that two types of cells are there. What are they? Sertoli cells are there and spermatogenic cells are there. So as I said, Sertoli cells are in supporting role. Okay, and spermatogenic cells means various stages of spermatozoa are present. That means spermatogonium may be there, then primary spermatocyte, secondary spermatocyte, spermatozoa, spermatids, all those things are present in the various part of the seminiferous tubule. Okay. Now each seminiferous tubule approximately 50 to 60 centimeter in length, and if you see the diameter 150 to 200 micrometer. Now the Sertoli cells which are present, they are also called as sustentacular cells. Okay, they are supporting cell. If you see the shape of those cells, they are columnar or polyhedral. Okay, and having euchromatic nucleus. Next, have you heard about blood-testis barrier? Anyone? Blood-testis barrier. This is very important. Okay, blood-testis barrier. It is very important. Why it is important? First, you have to understand how the blood-testis barrier is formed. So, I have told you that Sertoli cells are there. Now, suppose one Sertoli cell, another Sertoli cell, they are joined together, okay, with the help of junctional complex that is forming the blood-testis barrier. Okay. Now, that isolates the haploid germ cell from immune system, and that is called as secluded antigen. Now, what is the meaning of this? That means the spermatogenic cells which are developing, okay, inside the seminiferous tubule, where the various stages of development is taking place, okay, that is not exposed to the bloodstream. Okay, it is present inside the body, but it is hidden from the bloodstream. So as a result, what will happen if it goes into the bloodstream? It will be treated as antigen and antibody will be produced against it. Okay, so those kind of antigen which are present in the body, but it is hidden from the bloodstream, that is called as secluded antigen. Okay, so the spermatogonia or the stages of spermatocytes which are taking place, that is not exposed to the bloodstream, or else what will happen? Antibodies will be produced against it. Okay. Next, the Sertoli cells which are there, okay, they are having receptors, very important receptors. They are having FSH receptor on their surface. Now this FSH, that is follicle stimulating hormone. Now this follicle stimulating hormone, what it will do? It will cause or it will activate the cell in order to produce anti-Mullerian hormone. Anyone have you heard about this anti-Mullerian hormone? Anyone have you heard about this? You have heard about testosterone, you have heard about FSH, LH, all those things you have heard. But I don't think so you have heard about anti-Mullerian hormone. Anyone? No. तो क्या होता है एंटी मुलेरियन हार्मोन? Have you studied about the development of female reproductive system? Yes. What about the Mullerian ducts? क्या function है Mullerian ducts का? Most of the female reproductive system is developed from Mullerian ducts. Okay. Now this particular Mullerian ducts are present in case of male also, but their development is regressed in case of male. How? By this hormone. Okay, so this anti-Mullerian hormone, what it will do? It will inhibit the growth of the Mullerian duct in case of male. Okay, as a result, it will not grow. If it is not growing, then the parts of the female reproductive system will not be present. Okay, so this is very important. Okay, that role is performed by the Sertoli cells. Next, you can see there are other functions also of the Sertoli cells. First of all, secretion of androgen binding protein. Okay, it will help to bind the increased concentration of testosterone to the seminiferous tubule, which is required for spermatogenesis. Then secretion of inhibin, that inhibits the FSH release from the pituitary gland, anterior pituitary gland. Okay, then supports and nourishes the germ cell, and phagocytosis action is also there, which is required, okay, for the residual bodies of the spermatogenesis. Now here you can see the ultrastructure of the Sertoli cells. Okay, here you can see the ultrastructure of the Sertoli cell. Now see, these are the Sertoli. This is one Sertoli cell. This is one Sertoli cell. Okay, so the junctional complex which is present in between the Sertoli cell. Okay, तो Sertoli cell कहाँ पे रहता है? It is present inside the lumen of the seminiferous tubule. Seminiferous tubule के अंदर क्या रहता है? What is present inside the seminiferous tubule? As I said, only two types of cells are there: Sertoli cell and spermatogenic cells. Okay, so here you can see the interior of the seminiferous tubule. Inside the seminiferous tubule, first of all, you can see these are the Sertoli cells. So this is one Sertoli cell, this is one Sertoli cell. In between both the Sertoli cell, can you see the junctional complex? See, junctional complex. Now this is forming the blood-testis barrier. Okay. Now the junctional complex where it is present, okay, the lumen is divided into two compartments. Can you see the compartment? So the upper compartment is called abluminal compartment, lower compartment is called as the basal compartment. Two different types of cells are present over there, if you can see in the diagram. Okay, so if you see at the lower compartment we are getting the spermatogenia, and on the upper compartment we are getting the primary spermatocytes, secondary spermatocytes and spermatids. Okay, so like these two different compartments and different types of cells are present. That clearly shows that it is providing nourishment to the spermatogenic cells, as well as it is also protecting the spermatogenic cell, okay, by forming the blood-testis barrier. Okay, as you can see the blood vessel which is present. Okay, so it is forming the blood-testis barrier which is protecting the spermatogenic cells from the bloodstream, because it is not exposed to the bloodstream. As a result, what will happen if it is getting exposed to the bloodstream for the first time? The blood immune system will produce antibodies against them, okay, which will be harmful. That's why blood-testis barrier is very important. Now, when you will see the slide of seminiferous tubule, okay, when you will see the slide of seminiferous tubule, at that time, as I said, two types of cells you can expect. Now, if you see the entire lumen of particular one seminiferous tubule, at the basal part and at the upper part, what different types of cells you can expect, or how you will know where the Sertoli cells are present and where the spermatogenic cells are present. How you will know? That means what is the orientation of the cells. Okay, for that you can see the flow chart. So spermatogenic cells, they are derived from primordial germ cell. They replicate to form various stages of spermatogenesis, as you know. Okay. Now the arrangement, okay, of the cells, they are in stratified manner. Okay. Now what is the meaning of this stratified manner? When you will see the seminiferous tubule under low magnification, it will appear as stratified layers. Why stratified layers? Because different types of cells are present. Spermatogenia will be there, primary, secondary, spermatocytes will be there, spermatids will be there, Sertoli cells will be there. So they are in multiple layers. That's why it will appear that it is a stratified epithelium, but exactly it is not a stratified epithelium. Okay, so that you have to know. Next, if you see the arrangement from the basal lumina to the luminal area, that means from the basal part of the lumen to the upper part of the lumen, how the cells are arranged: first spermatogonia, then spermatocytes, spermatids, and spermatozoa. This is the arrangement. Okay, so this particular thing you have to follow when you are looking at the slide. And if you see the Sertoli cell, they are present at the basal part only. Okay. Now, in a particular cross section of the seminiferous tubule, all the cells may not be present. Okay, it is not necessary. It is not necessary that in any section of the seminiferous tubule, all the cells will be present. Next, a small amount of connective tissue is present around each seminiferous tubule, and that is called as lamina propria. Okay, where fibroblast cells are absent. Now, this particular lamina propria which is present around the seminiferous tubule, okay, this is a vascular connective tissue, and in that some amount of myoid cells are present. Rhythmic contraction of those particular cells helps in expulsion of the spermatozoa. Okay. Next, Leydig cells are there, or interstitial cells of the Leydig. Where they are present? They are present in that particular connective tissue. Okay. So the seminiferous tubule, cut section of the seminiferous tubule, as I said, they are surrounded by lamina propria, that is the vascular connective tissue. In that vascular connective tissue, these interstitial cells of Leydig are present. Okay. Now already you know that the function of the interstitial cells of Leydig, they produces testosterone. So this is the microscopic structure of testis. What you can see in this particular diagram. Can you see the layers first? I hope you can see the tunica albuginea. See, this is the tunica albuginea. Then this is the tunica vasculosa, which is the vascular cord. After that, what are these? These are the cut sections of the seminiferous tubules. Okay. So you will be getting multiple seminiferous tubule in the cut section. So here you can see these are the cut section of the seminiferous tubule. Now inside the seminiferous tubule, the white space which you can see, this is nothing but the lumen of the seminiferous tubule. Next, here you can see different types of cells. Here you can see different types of cells. In order to see those cells, you need high magnification. In low magnification, you can't see. You can see cells are there, but you can't differentiate. Okay. So these particular cells, these are called together, they are called as germinal epithelium. Okay, they are called as germinal epithelium. So, how many types of cells are there? Two types of cells. What are they? Spermatogenic cell and Sertoli cell. We are discussing about the seminiferous tubule. Several times I have mentioned, inside the seminiferous tubule, two types of cells are there. What are they? Sertoli cells and spermatogenic cell. Together it is called as germinal epithelium. In low magnification, you can only see the germinal epithelium. You can't differentiate the cell. In order to differentiate, what you have to do? You need high magnification. Is it clear? Now here you can see histology of testis, especially the seminiferous tubule under high magnification. What you can see over here? क्या दिखाई दे रहा है? See, first of all, you have to locate one. This is one seminiferous tubule. See, this will be another seminiferous tubule. This will be another seminiferous tubule. Okay. Now, suppose this is one cut section of seminiferous tubule. This is another. In between this, this particular area of connective tissue is called as lamina propria, which I have mentioned just now. Okay? If you see the interior, you can see different types of cells. So first of all, you can see the Sertoli cell. As I said, Sertoli cells will be present at the basal part of the lumen of seminiferous tubule. Okay. Next, if you go from basal part of the lumen to the upper part of the lumen, then you can see the different types of cells. You can see the secondary spermatocyte, you can see the spermatogenia. Okay. So the immature cells which are there, it will be present at the basal part. As it is becoming mature, it will go towards the lumen. Is it clear? So Sertoli cell and the immature cells, okay, they will be present at the basal part. Once it is getting mature, it is going towards the lumen. As simple as that. Okay. Next, can you see the Leydig cells? See, these are the Leydig cells. These are the Leydig cells. So where those Leydig cells are present, they are not present inside the seminiferous tubule. Okay. In between the seminiferous tubule, the connective tissue which is present, in that connective tissue only the Leydig cells are present. Function kya hai? To produce testosterone. Okay. Now these Leydig cells can also be identified. Okay. Under high magnification of testis, the Leydig cells can be identified easily. Now this is the schematic diagram. Now this schematic diagram you have to draw. Okay. So here you can see the tunica albuginea. Here you can see the tunica vasculosa. Okay. The layers. Can you see the layers? So this is the albuginea. This is the vasculosa. Okay. After that, what are these? These are the cut sections of seminiferous tubule. Inside that, the cells which you can see, these are nothing but the germinal epithelium. Okay. So this is under low magnification and this is under high magnification. Okay. So under high magnification only you can differentiate the cells. I hope it is clear about the testis. Next, after testis, what is the next part? Epididymis. Just now I have shown you in the diagram. From testis, whenever the production of the different stages of spermatogenesis is taking place, after that where it will go? It will go to the epididymis. Epididymis ka function kya? What is the function of the epididymis? Anyone? See, production is taking place inside the testis, especially in the seminiferous tubule. After that, what is the function of the epididymis? Storage. Okay. After that, the development. After that, the maturation, capacitation. Okay. The motility. Okay. All those things are taking place inside the epididymis. So what do you think, what you can see inside the epididymis? Okay. So in case of epididymis, two types of cells are there. Okay. First of all, if you see the lining epithelium, now you have to understand what is epididymis. Okay. If I show you the microscopic structure of epididymis, most of the students used to get confused. Okay. Most of the students used to get confused because in the epididymis I have mentioned that those efferent ductules, okay, they at the upper pole of the testis, the head of the epididymis, then those efferent ductules, it will converge into one highly coiled duct, and then it continues as body and the tail of the epididymis. That means you are expecting that when we will take the section, only one rounded structure will be seen, but it is not like this. Can you see the epididymis? See, these are the efferent ductules, okay, which is forming the head of the epididymis. Then from the head of the epididymis, there is only one highly coiled tube which is forming the body and the tail. And after that, it is continuing as vas deferens, as you can see in the diagram. That means what you will expect. If we take a section, only one cut section should be there. And that means only one rounded section should be there. But exactly it is not like this. Now this is the histology of epididymis. It is also appearing as seminiferous tubule. Can you see? इसका भी appearance seminiferous tubule जैसा है. Now you will ask me that there is only one tube. Then why multiple cut sections are shown over here? Why? What is the justification behind that? Have you understood? समझ में आ रहा है now what I am asking? Then why it is like this? Because here the section is taken at the head of the epididymis. तो head में क्या रहता है? Multiple efferent ductules are there, so those are also coming into the picture. And in most of the cases, the slide is prepared like this, not at the tail or at the body. It is taken at section where it is taken at the head level. So in head level, what we are getting? We are getting the efferent ductules. So those efferent ductules, numerous efferent ductules are there, approximately 30. Okay, so if you are taking the cross section, you are getting multiple rounded structures, as you can see in the diagram. Okay. Next, what is the differentiation? Next differentiation is inside the seminiferous tubule. What we have seen? Germinal epithelium. That also two types of cells. What are they? Sertoli cells and spermatogenic cell. But here, what we can see? Can you see the lining epithelium? ये देखो. This is the lining epithelium. Now in the lining epithelium, it is pseudo-stratified columnar epithelium, that also stereocilia is present, which were absent in case of seminiferous tubule. Okay. Next important point, if you see the pseudo-stratified columnar epithelium, okay, though it is pseudo-stratified, that's why the shape of the cells are also different. So it is having principal cells which are tall and basal cells which are short. So see the magnified image. You can see the principal cells, see the taller cells, and you can see the short basal cells. Next, the tall principal cells which you can see in the diagram, they are having stereocilia. What is stereocilia? These are nothing but the modified microvilli. Stereocilia nothing but modified microvilli. इसका फंक्शन क्या है? एब्जॉर्प्शन. टेस्टिस से जो प्रोडक्शन होता है when it reaches the epididymis, along with that fluid content is moved, so that fluid is being absorbed, and that is done by the stereocilia. That's why stereocilia is present. Okay, so I hope you can now differentiate, or you have understood the differences between the seminiferous tubule and epididymis. So in case of epididymis, the epithelium, what is the epithelium? Pseudo-stratified columnar epithelium. In that, two different types of cells are present. That is the principal cells and the basal cells. Principal cells are the tall cells. They are having stereocilia. And short cells are the basal cells which are not having stereocilia. Okay, that is the lining epithelium. Goblet cells are absent in this particular case. Next layer which is present, that is the muscle layer. Only one layer of circular smooth muscle cells are present. Okay, and gradually the thickness of the epididymis, means as you go downwards from body tail, okay, the thickness of the epididymis increases. Okay. Now here you can see the diagram under high magnification. So in the high magnification, what you can see? So see, these are the pseudo-stratified ciliated columnar epithelium with stereocilia, which you can see. In the lumen, mainly the sperms are present, okay, which are mature. Next, if you go outer to this particular epithelial layer, then you can see the circular smooth muscle layer. See this one, okay. So only one layer of circular smooth muscle cells are present. Okay. So this active contraction will help in expulsion of the sperms which are present inside the epididymis. Okay. After epididymis, what is the next part? Ductus deferens or the vas deferens. Okay. So if you see the ductus deferens, from where it is continuing, from where it is arising from the tail. So tail will continue as vas deferens or the ductus deferens. Okay. So it is a fibromuscular tube. So the ductus deferens or vas deferens is a fibromuscular tube. What is the length? Approximately 45 centimeter. Okay. So it will convey the spermatozoa from epididymis to the ejaculatory duct. Can anyone tell me how the ejaculatory ducts are formed? How the ejaculatory ducts are formed? Anyone? Okay. Now the end of the ductus deferens is a enlarged part that is called the ampulla. Okay. Now the spermatozoa transported along the vas deferens by muscular contraction. Okay, which are present. That means the smooth muscle cells which are present in the wall of the ductus deferens. Let me show you the diagram. Now in this particular diagram, can you see what is this? So see, this is the vas deferens. You can see this is the vas deferens. Now this vas deferens at the terminal part, it is dilated to form the ampulla. Next, the seminal vesicles which are present. Now the duct of the seminal vesicle and the vas deferens, they will join together to form the ejaculatory duct. And this ejaculatory duct, where it will terminate? It will terminate in the prostatic part of the urethra. Can you see the bladder? Now this is the prostatic part of the urethra. Now in this particular part, it is terminating. Okay. So I am talking about this particular duct, which is also called as vas deferens. The length is approximately 45 centimeter. Okay. Next, if you see the vas deferens, first of all, it is a fibromuscular tube. Okay. Now, if you take the cut section, so it is a tube shaped. If you take the cut section, what do you think? What will be the appearance? It is not circular. It is star shaped. Okay. Fibromuscular tube. If you take the cut section, what will be the appearance? Star shaped. Okay. So the lumen is star shaped. Next, mainly we are getting three layers. What are those layers? First will be the mucosa, where the lining epithelium is present. What is the lining epithelium? It is pseudo-stratified columnar epithelium with stereocilia, and small amount of lamina propria is also present. Okay. What is the lining epithelium? Same, almost same like the epididymis. That is pseudo-stratified columnar epithelium with stereocilia. Next, lamina propria. This is some amount of connective tissue is present. Next, smooth muscle coat. Now, smooth muscle coat is highly developed. The smooth muscle coat which is present in the vas deferens, it is highly developed. Why it is highly developed? What is the function of vas deferens? To carry the sperm. So if the smooth muscle coat is not developed, then how it will carry the sperm? Because the contraction of the smooth muscle cells are very important. Okay, that's why the smooth muscle layer is well developed. Okay, so in case of smooth muscle cells, three different layers are present. Okay, that means smooth muscle cells are arranged in three different layers. So inner longitudinal, outer longitudinal, in between these two longitudinal, a middle thick circular layer is present. Okay. Next, adventitia, which is made up of loose areolar tissue where blood vessels, nerves are present. Okay. Now, what is the function? Peristaltic contraction, which helps in ejaculation. Now, here you can see the diagram. In this diagram, I hope you can see this is the star-shaped lumen. Then this is the lining epithelium. As you can see, pseudo-stratified columnar epithelium with stereocilia. Okay. Next, small amount of lamina propria is there, and after that you can see this is nothing but the muscle coat. Okay, so in muscle coat you can see this is the inner longitudinal, this is the outer longitudinal, and between in these two longitudinal a middle circular coat is there, which is very thick. Okay, and externally we are getting the adventitia, where loose areolar tissue is present, mainly blood vessels, nerves are present. Okay. So these are the layers of the vas deferens. Okay. So identification of the vas deferens is not that difficult, very simple. Star-shaped lumen will be there. In high magnification, you can see the stereocilia also, and along with that, three layers of smooth muscle followed by the adventitia. Okay. So that is all about the vas deferens. Okay. So few topics are left. As I said, whatever portions will be left, that we will discuss in the histology demonstration class. Thank you.